Thank you very much, Neil, and thank you to the Royal College for the invitation to present this lecture on this day, which is becoming a very important day in the Royal College calendar and in such impressive surroundings. I've never quite been surrounded by an audience before. What I want to do today is to talk about my early days in Kenya and Glasgow. Then when I moved to ICI Pharmaceuticals, about the discovery of Propofol. And the process was not straightforward, so we'll go back and forward a bit about the trials and tribulations there. A lot of it leading to the need for new formulations, and I'll say a bit about the development of new technology that the profile of the drug allowed. This is the farm in Arn where I grew up, in the distance here on the side of the hill looking across to the Mull of Kintyre. And I decided to study veterinary medicine at Glasgow. And I presume my expectation at that time was that I'd be driving around the countryside doing large animal practice. But after graduation, I decided that I would like to continue in the academic environment. And unfortunately, the role that I would have liked, house surgeon in the surgery department, had already been allocated. So I went to see Professor McIntyre, and he said, yes, you can have a job in medicine, or you can come to Kenya for a year. So I said, right, I'll come to Kenya for a year, where a group from Glasgow were setting up a conversion course for East African diplomates who were being converted to a degree course in Nairobi. So before I went, I did a couple of locums in one-man practices, and in Butte, I had my first experience of a cow with a prolapsed uterus, and in Dunoon, another one-man practice. The owner held the dog while we anesthetized it with pentobarbital for a hysterectomy and a pyometra. So on my own, I did manage both of these, but that was fairly my kind of limited experience in general practice. So off we went to Kenya. This is Professor McIntyre at the end, myself, Max Murray, and Douglas McEwen. And there, my role was to be involved in an ambulatory clinic in the local Kikuyu Reserve, where we treated animals or selected suitable cases to be brought in to the hospital at Kabete for examination by the students. And I had a couple of assistants who would translate the Kikuyu symptoms for me. And many of the cases were cattle, and the three common things were anaplasmosis, which was treatable, 3-day sickness, which they would get better on their own, although they looked very ill, and East Coast fever, which was invariably fatal. And the owners, I had a very polite letter from one of the owners of a cow that had East Coast fever, and it said, "Dear sir, I have the honor to inform you that the cow you treated yesterday has died." So after a year in Kenya, I heard that I was able to get the job that I had sought at Glasgow in the small animal surgery department. And rather than a 3-week trip by boat, as we had in the way out, I flew back and joined the surgery department in Glasgow. There, I developed an interest in dogs with sublingual mucus eels. These had originally been thought to be branchial cysts and required a fairly extensive total dissection, which was very complex. and difficult with all the major blood vessels in the area. More recently, at that time, it had been thought they were related to some abnormality of the salivary glands or ducts. And I began to examine a series of these and explored silography of the sublingual and submandibular ducts. And this is a picture of a normal sublingual gland and duct. The duct is coming along there. Then there's the anterior and the posterior part of the sublingual gland lying in the same capsule as the mandibular gland, which is just behind it. So I did mandibular silograms as well. And this is the silogram of that dog that I showed you with the submandibular swelling. It also had a ranula, and the duct is fine coming along here, and then there's a leakage from the anterior part of the gland into the ranula, and from the posterior part into the cervical swelling. And it proved that simply excising the mandibular and sublingual gland was effective. And over my time at Glasgow, I was able to accumulate a series of about 50 of these cases, and I collected pathology from the glands and the cysts. And I had registered to do the reason I'm telling this kind of non-anesthetic story is that I'd registered to do a PhD on studies of salivary gland conditions in the dog. And many years later, when I had changed to do anesthesia at Glasgow, and I'd looked at a number of new drugs there, and then moved to ICI, I had a letter from the clerk of the faculty in Glasgow saying that they had noticed that I hadn't submitted my PhD, and that possibly they could consider a change of title. So I was delighted that they were able to change it to studies of intravenous anesthetics in animals. The work I did at Glasgow that related to anesthesia, I'd been fortunate to obtain the Diploma in veterinary anesthesia that had been introduced just at this time. I was one of the first to take the examinations for the diploma, and I was doing all the teaching and research in veterinary anesthesia. And at that time, there were a number of new drugs available to experiment with, and most of my time was done with the neuroleptalgic combinations, with ketamine, and also validating end-tidal CO2, prediction of arterial CO2, and indirect blood pressure measurement, primarily in dogs, but we also did some in horses as well. And it was this interest in the research side, looking at these new drugs, that stimulated me to apply or to respond to an advertisement from ICI Pharmaceuticals looking for somebody to come and do research in anesthesia. I was successful and headed down to Alderley Park in Cheshire, and it's a lovely greenfield site with a whole range of buildings and all the facilities for research there. The supply of animals, the chemists were there, the pharmacokinetics people were there. Everybody was in hand to give you the help that was required. And the anesthetic projects team that I joined had very simple objectives: to search for new anesthetic drugs and to evaluate new methods of looking at anesthetic drugs. And ICI Pharmaceuticals had a history in this area with the discovery and the introduction of halothane, and there were people working in the lab who had worked with Raventos in the days of halothane's discovery. We did look for new inhalational agents, but didn't find anything there that was going to be sufficiently interesting to take forward. I did, I admit, examine sevoflurane as a licensing in compound that was presented to us as an option, but and it looked a very good anesthetic as far as its anesthetic properties, but we could see that it was very unstable to soda lime, and that when we put it in a flask with soda lime, it just disappeared over a relatively short period of time. And the breakdown products were believed to be nephrotoxic materials. We did a study in dogs which showed that we could measure this breakdown material. It didn't appear to accumulate over time, but I was concerned that this material, this compound, although it was a good anesthetic, was too unstable in soda lime to take forward. The Americans came to the same decision, but Maruishi in Japan took a gamble and got away with it. So the focus for me was primarily on intravenous agents, and thiopentone had been the gold standard since the 30s because primarily of its rapid, smooth, the quality of anesthesia it produced, the rapid, smooth onset, and the lack of excitatory side effects. So many new agents come along and they're promising initially, but then they find that the patients say, well, they're twitching a bit. You know, it's the quality, the smoothness of anesthesia that thiopentone produced. But there are some downsides if it's given for any length of time. Repeated injections lead to quite prolonged recovery. And although the initial recovery is rapid, there is some post-operative drowsiness. So the target that we were looking for was a drug that would mimic the quality of anesthesia that thiopentone produced, but would provide the opportunity for use by continuous infusion for maintenance of anesthesia or for sedation, because it would be much more rapidly metabolized. And that was the basic target that we set ourselves. It had to match thiopentone, but add something to it. The catch-22 with looking for new anesthetics is that for a drug to get across the blood-brain barrier, it has to be a lipophilic compound. And if you have a very lipophilic compound, then it's not going to be aqueous water-soluble. And that meant that thiopentone crosses the blood-brain barrier because it can be achieved in a sodium salt, and it dissociates to the active base, which crosses the blood-brain barrier. But it meant that many compounds that might have been tested for anesthetics were not, because they couldn't be obtained in solution. And then along came, in 1966, propanidid. And propanidid was formulated with this surfactant, cremophor EL. And cremophor allows the formation of an aqueous looking, it's a clear micellar solution. And cremophor has a core of racinoleic acid. It's a lipophilic core that allows the lipid type of drug to be dissolved in the core, and the polyoxyethylene side chains provide water solubility. So the availability of cremophor as a vehicle allowed us to consider testing drugs that we had in our compound collection at ICI that hadn't been tested as anesthetics before. If we look at the options that we have for looking for new drugs, the chemists we had in the team, some of them were looking at modifications of existing drugs. You know, could you improve on ketamine? Could you improve on etomidate by making minor changes? For a rational design of a new drug, then you need to know something about a receptor that you're aiming for, and that's the case with beta blockers and things of that sort. A number of drugs have come from natural products, the belladonna alkaloids, digitalis, and these really were ongoing. We would look, if anybody came across with these and wanted to test them, we would test them. But what one of our chemists decided was to go back to the old compounds that ICI had in its compound collections, and that included not just the pharmaceutical division, but also the organics division, petrochemicals. They might have had something that could be of interest if it was tested as an anesthetic. And Roger James selected a number of these old, untested lipophilic agents, and the primary screen that we had, they were dissolved in cremophore, and I'll show you just the setup in a minute, but we looked for the median hypnotic dose, the dose which would produce loss of writing reflex for a 30-second period in 50% of mice. And the median lethal dose, that would now be the maximum tolerated dose. And the ratio between the two gives an indication of the therapeutic ratio, the safety margin between the two. So you want the widest margin between the two as possible. And this is the setup that we use in the primary screen. We have the mice in this Perspex holder here. There's a slot along the top, so you can hold it by its tail and slide it in here. And the veins in the tail are quite easily visible. It's easier to give injections to the mouse than it is to the rat. And this mouse has actually fallen asleep. Its head's gone down. And the lead compound that we found was 2,6-diethylphenol. This was the first compound in this batch of previously untested old compounds that we came across. And when there's a lead, we say it's a lead because it doesn't fulfill all of the requirements. It wasn't particularly potent and not particularly rapid in its onset. But the chemist then can say, right, this is showing us the direction we should be going. What have we got that looks related to this? And Roger James then went back to the collection and got other related compounds. And he also synthesized quite a number. I think we looked at about 300 related alkyl phenols by the end of this part of the process. This shows the laboratory record of the first day that the anesthetic effect of 2,6-diisopropylphenol, the one that became Propofol, was observed. And that was on the 23rd of May, 1973. And you can see that a dose of 100 milligrams per kilo was fatal. So we came down to 10. They were very rapidly going to sleep, immediate side position. And with 5, they weren't asleep at all. They were just slightly ataxic. So the minimum, the hypnotic dose was down between 5 and 10 milligrams per kilo, and then we looked at larger dose again to get up to a median lethal dose, and they established the therapeutic ratio. So this is the structure of Propofol. It's a hindered phenol, which means that the hydrogen bonding properties of natural phenol on its own are reduced by the effect of the side chains on either side. And it seemed important that of all the phenols we looked at, this was the one that had the optimum balance of properties. We had a number of secondary tests, But one of our most important tests was, a lot of compounds would pass the primary screen and they would look good in the mice, but then in the secondary test, most of them fell down. And the rabbit is a remarkably sensitive species to compounds that are likely to produce any degree of central excitation. And many compounds that may look quite good in the mice were excluded because of the excitation that they produced in the rabbit. So the rabbit was an important secondary test for us. If they looked good in the rabbit, if the onset of anesthesia was rapid and smooth, and the recovery was smooth, then that was them looking promising. A number of new drugs have come along recently, and there was an etomidate analog not so long ago that got as far as clinical trials. They must have spent a lot of money on it, but it failed in patients because of the amount of excitation that was produced. And I believe if they'd looked at the rabbits, they might have saved a lot of money. So speed of onset is also important for a drug that's being used for induction of anesthesia. And we did quite a bit of work just establishing our methodology here, because to compare speed of onset between different agents, you have to make sure that you're giving equipotent doses over the same time. If it's something is less potent and you take longer to give it, then you can't really compare that with something that's more potent and you give it more quickly. And this is just a few of the alkyl phenols to show why we selected the isopropyl. This is the isopropyl here in slightly heavier type. This was the lead compound, but you can see it's a lot less potent than the isopropyl. It was reasonably rapid in onset. Again, the dipropyl, less potent, slower in onset. This one here becoming more potent, but getting slower in induction time. And an isomer of this one was looked at again by somebody recently, but has not been taken forward. The lipophilicity of the side chain increases as you go from the isopropyl to the tertiary butyl, but by that time then it's almost inactive, and it must be it's not just lipophilicity, but possibly the size of the molecule has determined that this is too big to get across the blood-brain barrier. So this was the selection of the 2,6-isopropyl phenol. To assess recovery, we compared Propofol with thiopentone, and we gave equipotent doses. I think it was 2 times the minimum hypnotic dose, and we looked at the duration of sleep. We looked to see how quickly they walked when they recovered their writing reflex, and to get an indication of how fully they had recovered, we asked the mice to balance on this rod, a very complex test, isn't it? So they had to either balance on the rod for 20 seconds or walk to the end of the rod in a shorter period. And if we look at the results, the sleeping time, thiopentone mice were sleeping a little bit longer, but both groups, the thiopentone and the propofol mice, as soon as they regained their writing reflexes, they were walking away. And they looked as if they'd recovered, but when we asked them to balance in the rod, the propofol mice could do this within about 3 minutes, but the thiopentone mice were still falling off after about, it took 50 minutes until they were able to do this without falling off. So it was a clear demonstration of the complete early recovery, the clear-headed recovery with propofol, and the hangover effects with thiopentone. And the other study that was showing the key differences between the two drugs is that we looked at accumulation after repeated doses. And with the first dose, propofol and thiopentone produced about the same short sleep time. But as we gave thiopentone by repeated doses, the sleeping time got longer and longer until they were sleeping for 3 hours, indicating the accumulation of effect that was occurring with thiopentone. Whereas with propofol, we were able to give 10 doses with only a very marginal increase in sleeping time. So this is the lack of accumulation, which potentially suggests that the drug would be suitable for administration to maintain anesthesia by repeated injection or by infusion, whereas that's not really a possibility with thiopentone. The next thing we did was to look at the effects of propofol and compare it with thiopentone and althecin in a larger species. And normally we would use dogs and examine the hemodynamic effects in dogs. But cremophore, dogs don't like cremophore. This is the effect of a single dose of cremophore in a dog. They get a marked hypotensive effect on a first administration. They're just very sensitive to the effect of cremophore. They get histamine release. And so we had to use pigs, and found that working with pigs was actually quite nice. My assistant became very expert at anesthetizing pigs and inserting cannulae in a carotid vein and in a carotid artery and a jugular vein. And that allowed us to administer drugs without having to restrain the animal and to record arterial blood pressure and heart rate. So we wheeled the animal along to the lab in this box, connected them up, and there's a transducer down here. So after the pig has lost consciousness, its heart will be about the height of the transducer down here. And we were able to demonstrate that the effects of propofol were really quite similar to thiopentone and althecin in the pig, and certainly were acceptable. Where do we go now? So the other thing that was going on in parallel with this detailed pharmacology was that the safety of medicines people have to evaluate repeated administration. They did a 30-day study in rats, 30-day study in pigs, and I take my hat off to them, anesthetizing pigs every day for 30 days. But they achieved all that, and they looked at mutagenicity and all the various things that have to be done. And a whole package of data has to be submitted to the Committee on Safety of Medicines before clinical trials can begin. All of the animal safety data, all of the pharmacology, and the pharmaceutical information about the formulation. And it takes quite a while for that to be assessed. And while we were waiting to get approval to begin trials, one of our medical information officers had a friend who was an anaesthetist in Derby, and he said, well, I'm going to do a sabbatical in Belgium soon, and Professor Raleigh, I think, will allow me to do some trials as long as you can assure me that all the safety information has been obtained. So this seemed like a shortcut at the time. We didn't have an anaesthetist in the company on the clinical research side, so we had a cardiologist that was the link man, and off Dr. K went to Ghent in Belgium with some samples that had been provided for him. And I've got a few tables in this presentation because if a fairly major thing happens in your life, you remember where you were at the time. And I was at a development team meeting that we had from time to time, and this was 3 days after Brian Kay had gone to Ghent, when a phone call came through to say that he'd studied 22 patients already. And I said, what? And he said, yes, it's an anaesthetic, but it's more potent than you think. And because Professor Rowley was editor of the Belgian journal, he was able to get a publication out fairly quickly. And here he says, in 22 unpremedicated patients, the dose was 0.94 milligrams per kilo, about half of what I had expected. And he also said there was pain on injection in some patients, which I hadn't seen in the animals. We certainly saw no pain in mice, in pigs, in dogs, in cats, in monkeys. And only later did we look at our rats again, and we had seen a bit of struggling response in the rat when we gave the injections, but we presumed it was just down to the restraint that was required to give them the injection. But when we gave some fentanyl prior to Propofol, we abolished that struggling response. So the rat was the one species of animal that we could see some pain on injection. So rat and man seem to have something in common, and I believe they have some kinin precursor that is triggered by Propofol, that a cascade goes down and eventually produces a painful kinin that produces pain. And it seems to be very much species specific, and not all patients and not all rats have the necessary precursors to induce pain. So having heard that there was pain in injection and that the potency was more than we had expected, we reduced the concentration from 2% to 1%, and we took out the alcohol for the formulation because we'd added some alcohol to get a totally clear solution. And studies then continued, but they continued, maybe I've got a little, what have I got? Yes, by this time we had a psychiatrist in clinical research, and he was handed Propofol next. To me, it's a little bit like asking a learner driver to demonstrate a Formula One car, but this is what we had. I tried to encourage the manager of clinical research to employ an anaesthetist. And he said, well, the next time we're advertising for a cardiologist, we'll ask for one with an interest in anesthesia. And that's as far as we got. But so this psychiatrist said, we'll do a double-blind study against althecin with the dose of Propofol that Dr. Kay had determined, and we'll compare it with althecin. And after a few days, we heard that the patients were not falling asleep. One particular patient in Glasgow was lying there, and he said, when does this stuff reach my brain? So the studies had to be abandoned, and we had to start dose-finding studies again. And the dose did turn out to be much nearer to 2 milligrams per kilo, as I had predicted. And only many years later did I find out why this mistake had occurred, because I was interested to find out which patient was the first patient to receive Propofol as an anesthetic. And there was a problem in the records that came from Dr. Kay were a single page with the patient's age, weight, the dose given, and a little bit about blood pressure measurement. But compared with a clinical trial nowadays, where you got a great book of data to collect, it was very limited. But patient number 1, and that was dated the 15th of February. There were quite a few were the 15th of February, but patient number 3 was the 14th of February. So I went back to Professor Rowley many years later and said, is it possible to find out which patient first received Propofol? Professor Dundee's book has got a picture of the record of the first patient to receive thiopentone. And I thought, we must have something similar for Propofol. But so he came back and said, actually, it is number 3. And he also sent me the hospital record of patient number 3, which made no mention of Propofol, but the patient had been pre-medicated with 2 mils of thalaminol, droperidol and fentanyl, and had then gone on to have an 8-hour operation for excision of a facial tumor with neuroleptic analgesia. So that would never be allowed nowadays, and it was a reflection of the lack of control that was allowed to take place in the early stages in Belgium. But it was this, I'm sure it was the fact that Dr. K didn't know that the patient had been pre-medicated or assumed that they hadn't, that he was probably just going on to a list and say, can I give a wee bit of Propofol before you do your operation here? And that's really what it looked like. And after he'd done his 5-minute measurements or so, he handed it back to the anaesthetist. So the induction dose does appear to be very close to 2 milligrams per kilo. Now, the progress of a compound in development, once it's been accepted as a candidate drug, then there's a development team with representatives from the major functions that are going to be involved in its development. And these include regulatory. I was the biology representative. Clinical research was our psychiatrist. Pharmaceutical people were important because we were already beginning to look at alternative formulations. The commercial people begin to say, well, what's the forecast? How important is this in our portfolio and our kinetics? And people are beginning to look at the distribution of the drug. And the development department coordinates it all. So again, I think I've got, these were, the follow-on studies had indicated what the dose was. The doctors involved and the anaesthetists involved in these were prepared to continue and do more studies, but they weren't overly enthusiastic because there was still some concern about Cremophor. The pain on injection obviously was undesirable. There would be a fairly prolonged development time, particularly if we did decide to change the formulation because of the question marks over Cremophor. While we would advocate its use for maintenance, there were no established techniques for maintenance. And our pharmacokinetic expert was not convinced of its likely utility for maintenance, looking at quite a long final elimination half-life. I had seen the animals recovering after infusion, and I was convinced it would be fine, but he wasn't so sure. And back to our table again, but I was sitting in a different place this time. I was round this side, and the chairman was here. And by 5 votes to 4, we decided at that meeting to continue development. There was a lot of negative comment, and it could have all halted at that time. But I certainly voted to go ahead, and the chairman voted to go ahead. I have the names of the others, but I can't remember how they all voted. So, as I said, there had been question marks about Cremophor for a long time because both althecin and propanidid were associated with an incidence of anaphylactoid type of reactions, and where the patient's blood pressure would collapse very suddenly, sometimes bronchospasm, other signs of anaphylaxis. And what we had seen when we were doing our hemodynamic studies, comparing propofol with thiopentone and althecin, there was one occasion when we gave a second administration of althecin that we saw what looked like an anaphylactoid type of reaction. In the patients flushed, and well, these are the signs that we see with these reactions that we get. And what we found was we set about to do a prospective comparison of the cremophore-containing agents with and without cremophore. We used a non-cremophore mixture of propylene glycol and alcohol and compared them with cremophore and with the non-cremophore formulation. And the features of the reaction that we found was that if we gave a second injection after 4 days, no problem. After 7 days or up to 14 days, we got this characteristic response. And if we went after 4 days, then after 2 weeks, or challenged them after having had a reaction, we didn't get a further reaction. So it wasn't a true anaphylactic response, which would have been exacerbated if you keep giving a further challenge. So it required this unique sort of dosage interval, and initially they get generalized vasodilatation with blotchy cyanosis, hypertension rather than hypotension, and I think this is a response of the pig to histamine, and there may be some noradrenaline release as well. But they certainly get histamine release. And this was a feature of the reaction, that very quickly there was a rapid decline in polymorph counts. And that is suggestive of complement activation with release of chemotactic C3a and C5a factors, which lead to margination of polymorphs to the sides of blood vessels, so that when you sample, you get a much reduced count. And it's a fairly transient effect, and it recovered after about 20 minutes. But that was certainly a consistent feature of the response. And a bit of an increase in sleeping time as well. And this was what the kind of blotchy cyanosis that we saw after the initial hyperemia phase. And this is a summary of the results in the mini pig, so that cremophore, back where we were. Cremophore on its own produced a high frequency of responses. Alcohol, propylene glycol was fine. Epontol in cremophore, high frequency of responses. Propanidid, the active agent in epontol, avoiding cremophore, avoided the reactions. Althecin and cremophore, high frequency of reactions. Alfaxilone, alfadalone on this non-cremophore formula, still producing some reactions. And I invited Ben Davis from Glaxo to come and see our results. And he had to accept that's really what we had found. And I suspect they may have repeated and got the same results themselves, because alfaxilone and alfadalone never appeared in an emulsion as it might have if they'd really wanted to continue with it. Alfaxilone, now reintroduced in veterinary practice in cyclodextrin, and I just wonder if it's used in man, whether this might be a problem again. But it's not released for use in man yet. There are still studies going on. So because of the concern about Cremophore, we were looking for another formulation. And to begin with, we looked at what other surfactants are there that we might use. And what we found was that a lot of the surfactants, the Cremophores and the tweens, all have some naturally occurring lipophilic core, racinoleic acid, stearic acid, and all of these were bad. They produced the same problem. But there were some surfactants that had a synthetic core, totally synthetic polyoxypropylene core that was able to absorb the lipophilic material. It had the same polyoxyethylene side chains, and these were tolerated at the same sort of doses as Cremophore, as in an acute toxicity test. They seem to be very safe. And that's the one that we selected, a synperonic, which was a compound that we were able to obtain from ICI Petrochemicals, and repeated all the pharmacology again, repeated all the toxicology, another 2 years of work there. But unfortunately, there were some histological changes. Although the material was tolerated well, the animals continued to put on weight and appeared to be fine from that respect. The histological changes in the liver were such that the pathologists were not happy to approve of this to go forward into patients. So that was 2 formulations down, and we then had a meeting back to the table again, and this time I'm up here and the technical director sitting at this side, and they accepted that synperonic was not a suitable formulation, but they then said, well, it might take a long time. We were beginning to look at emulsions at this time, and that seemed to be the answer. But because that would take quite a bit of time, the safety of medicines people said, well, we didn't see problems in pigs when we gave Propofol every day in the Cremophore formulation. I think you can go ahead and market the Cremophore formulation until the emulsion one comes along. And that was very much against my advice, and I was very concerned and unhappy about that decision. Studies went ahead, and certainly within a few months there were about 4 anaphylactic reactions reported with the Cremophore formulation, and studies had to be halted. So I was pleased we were back on track again, and we were back to the emulsion formulation. And we had looked at emulsion formulations quite early in the project because they seemed a rational way to go. But the emulsions then were such that the particle size probably wasn't small enough, and the effects of Propofol were not maintained. The onset of anesthesia was slower, the potency was less. They were just not satisfactory at that stage. But emulsion technology had improved. It was possible to get much smaller particle sizes. It was possible to get very stable formulations. And the one that we, after looking at many different emulsion formulations, the one that we came up with is this soybean oil, which dissolves the drug, and it's then added to an aqueous preparation that contains purified egg phosphatide, lecithin type material, and that coats the surface of the droplets to prevent aggregation and coalescence, and stabilizes the emulsion. And the particle size of the emulsion is about less than 0.25 micro, well, very small, very small. So again, we had to repeat all of the pharmacology again. We had to repeat all the toxicology, but we could use dogs on this occasion, so that was slightly easier. And we had to repeat all the clinical trials that we'd done, all the dose-finding studies, but we had a very efficient doctor join the team, Dr. Ron Stark, who had experience in respiratory physiology. And he, with the knowledge of what had gone on with the Cremophor trials, set up a very efficient and very quickly conducted study to repeat the dose finding, and then got the emulsion formulation approved for induction and short-term maintenance of anesthesia in 1986. So that's 13 years after discovery of the compound, because of the fact that we had 3 different formulations, 3 different sets of pharmacology and toxicology, and two lots of clinical trials. So we did have to try again there. That led on to expanding the clinical trial base to look at other indications, and long-term maintenance of anesthesia was one that I was very much involved with, and I'll say a little bit more about the technology that we came onto there. Also use for sedation for investigative procedures like bronchoscopies and colonoscopies, gastroscopies, and also for surgery done with local regional techniques where the patient likes to be sedated at the same time. Use in children came later on. Use in obstetrics, we never really got very far with that because some of the early studies used rather large doses and the APGAR scores weren't particularly good, but I think it's used quite a lot in obstetrics now because thiopentone in many places is difficult to get a hold of. We also had a 2% formulation, and this is something that I was very much involved in, which I'll say a bit about later. So the fact that we had a drug that could be given by continuous infusion for maintenance of anesthesia encouraged me to look at the practicality of this. And to give the drug for maintenance, you either use a syringe pump or a volumetric pump or a simple drip, but that's really not very accurate. The syringe pumps had very limited delivery rates at that time, and volumetric pumps were very chunky and expensive. So I approached a lot of manufacturers of syringe pumps to alert them to the opportunity here to produce something that would facilitate use of propofol for maintenance of anesthesia, and alert them really to the opportunity similar to the development of the tech vaporizers for the administration of the inhalational agents. And the first company that went to this was the Fresenius Injectomac pump. It had a maximum rate of 99 mils per hour, which really wasn't enough for a large patient. And they said, well, we can actually manage to extend it up to 150 mils per hour. So that was used in a number of studies for a time. How are we doing for a time? All right, so far. Then Omida came along, and they produced this new prototype that was tested in a couple of centers. And this had a number of extra features in that it could give a maximum rate of 200 mils per hour, and also bolus infusion rates such that the induction dose could be given by infusion. And that was something new and something which allowed it to be used for target-controlled infusion as well. Target-controlled infusion, just very briefly, requires a pharmacokinetic model and control algorithms to be installed within the pump. And what the anaesthetist does then is, rather than set an infusion rate, you set the target blood level that you want to achieve, and the pump calculates what the initial dose will be and then what the infusion rate needs to be to maintain that concentration. And you can titrate up and down the target as you want to increase or decrease the depth of anesthesia. And this is just an outline of the profile that you will achieve with target-controlled infusion, where an initial bolus is given rapidly, it's not shown here, and then the pump calculates a declining infusion rate over time to maintain the target concentration here. When you suggest a lower target, it switches off the infusion until the concentration is predicted to have fallen to the new target, and then it switches on again at a lower infusion rate. If you want to go back up again, it gives a bolus and then a faster infusion rate. So it makes it easy to titrate the depth of anesthesia by adjusting the target rather than setting an infusion rate in milligrams per kilo per hour. So overall, I think the benefits of Propofol are summarized here. We do achieve generally the rapid, smooth induction that we looked for. And the laryngeal mask, this was invented and introduced about the same time as Propofol. It's an alternative to the endotracheal tube, and it means that patients don't have to be paralyzed, and there's less likely of traumatic injury to the trachea. And Propofol and the laryngeal mask seem to go very well together. Propofol facilitates the introduction of the laryngeal mask, and both of these are very often used in outpatient anesthesia, where the swift, clear-headed recovery and the minimal nausea and vomiting is appreciated by patients. So these, I believe, are the main features that have made Propofol as popular as it is today. A recent survey in the UK said it was used in about 2 million patients in a year and used in about 90% of general anesthetics in the UK. So it's certainly well established and I think quite well used in veterinary anesthesia as well. I just finished by saying anesthesia is quite like flying, and the takeoff and the landing are the tricky bits, and that's a landing that I was fairly happy with. Thank you very much indeed.
Thank you, Ian. The point you made about the steroids producing a reaction, I mean, that does concern me. And do you think anybody's following it up at the moment?
Well, I mean, I know the Colin Goodchild, who is chief executive, I think, of the company in Australia that are doing clinical trials, but I haven't contacted him in any way to suggest that he might look at the pig model. Yeah, might be worthwhile. Might save him some money.
Thanks very much for your presentation. I just want to ask a question about the pain element, because I've had Propofol myself several times. The last time I was in excruciating pain. And when I worked for Fort Dodge, the company introduced a water emulsion of Propofol called PropoClear. And in all the early trials, they didn't really notice in America any significant pain. But when we introduced it into Europe, there was an incredibly high rate of pain on injection, so the product was withdrawn. Can you speculate on why it does cause pain?
Yeah, I mean, I think it's related to the free aqueous concentration. And I would imagine that the free aqueous concentration was higher in that clear formulation that you mentioned. As I say, there seems to be species differences, and I think there are interpatient differences. So some patients in my view, have the kinin precursors and some don't. So I've only had propofol once and I didn't feel anything. But, and some other people do feel certainly with, there is a delay. I mean, the reason I say I believe there's a kinin activation is that it's not immediate as you give the injection. It's usually as it travels up the arm. And I think there's a delay while a kinin cascade is triggered, in my view. And some patients have maybe more of this kinin precursor kininogen than others. It's readily controlled in patients if a patient is concerned about pain and injection, or if an anaesthetist is concerned, he can either mix some lignocaine with propofol before the injection is given, or give some lignocaine IV before the propofol. And that either, in the case of mixing it, I think it probably stabilizes the propofol kininogen interaction.
Certainly one time I was given fentanyl and
That worked too.
Any other questions?
I've been told that after on recovery of propofol, I don't think it applied to me when I had it, but it certainly applied to my working partner when he had it. But there was a period of hyperactivity after on recovery that you were sort of euphoric.
I think some people have reported a degree of euphoria, and I've heard of some people having quite amorous dreams as well, and things of that sort. But I was a bit involved in the trial of Michael Jackson, on the trial of Conrad Murray for the manslaughter of Michael Jackson. And one of the supplementary bits of information there, the doctor, the cardiologist, was claiming that we only gave him 25 milligrams of propofol, and he hadn't been doing this regularly. But 225 vials of propofol had been delivered to his girlfriend's address in Las Vegas, so that's been used for something. And I think Jackson was probably being given it every night for about 2 months, so he must have liked something about the experience of having propofol.
This is a slightly technical question. In your mini pig studies, you showed that there was peripheral vasodilation or generalized vasodilation, but also tachycardia and low blood pressure. And I was just wondering how you can get, was it high blood pressure?
There's hypertension, yeah.
How do you get high blood pressure when you've got generalized vasodilation?
Well, it may just have been a peripheral vasodilatation, just in the skin. I'm trying to think whether we did. Well, we certainly got increased histamine response, and I think in the pig, histamine, rather than giving hypotension, does give hypertension. Yeah.
Any other questions? Any other burning insight into the Michael Jackson investigation? Now's the chance.
I knew both of the expert witnesses, and Paul White emailed me a couple of months before the trial to ask what did I know about the oral absorption of propofol, the oral effect. And I was able to tell him that virtually nothing happens. As rapidly as it's absorbed, it's metabolized. And he apparently had suggested that Jackson might have swallowed some propofol himself as a defense strategy. And then Steve Schaefer from Stanford said a few weeks later, "You'll know that Paul is acting for the defence and I'm acting for the prosecution." And Paul hadn't mentioned that. But Steve Schaefer from Stanford was a fantastic expert witness. He was setting up drips in the courthouse and identified 17 egregious errors of standard of care. Lack of monitoring equipment, lack of resuscitation equipment. He was out on the phone half the time. He was found, Conor Murray was found guilty, sentenced to 4 years, and he was out after 2. Not enough.
Well, thank you very much for a wonderful presentation. We have a little gift for you.